Okay, so let's go ahead and get started. Welcome to the fourth and final joint EEB complex systems seminar. Amy Wesolowski is our fourth candidate for the theoretical ecology position that would be split between EEB and complex systems. Amy got her bachelor's degree in human ecology with mathematics at the College of the Atlantic, and then went on to a PhD. She applied here.
I don't know if I ever applied.
We expressed some interest here, but then went on with our loss, and she went on to Carnegie Mellon and did her PhD in engineering and public policy, getting her degree in 2014. And since then, she's gone on to do good things. She's was a postdoctoral fellow at Harvard Public Health for a couple of years, and has been at Princeton for the last few months. She has an impressive number of publications you see from looking at her CV. And some highlights there include a McDonald Foundation postdoctoral fellowship and Burroughs Wellcome Award she's recently started. So without any further ado, let's listen to Amy talk about the impact of human mobility on infectious diseases.
Thanks so much for having me. So as Aaron said, I'm Amy, and I'm a postdoc in EEB at Princeton. And I'm going to talk today about some of my work looking at trying to quantify human travel and relate that to infectious disease dynamics. So very broadly, in disease ecology, we're interested in trying to think about how pathogens are interacting with susceptible human populations and how that ultimately leads to a disease outbreak. Sort of what's driving those underlying dynamics are going to vary depending on what pathogen you're looking at and what environment. So pathogen dynamics can be determined by environmental drivers, different types of animal reservoirs, vector biology, heterogeneity within hosts, and evolution. And susceptible human populations, their underlying dynamics are also going to be determined by a lot of things. You can have an increase in births that increase your susceptible population. You can have host susceptible heterogeneity. You can have things like vaccination that changes your landscape of immunity. And what I'm interested in looking at is about mobility patterns and how that changed the susceptible dynamics. And so most of my research has primarily focused on trying to look at human populations for a range of different types of pathogens and trying to see sort of what commonalities can we see about human behavior and dynamics. And in particular, how we can think about the interactions between the environment, vector biology, pathogens, and human populations. And most of my research uses a range of mathematical, statistical, and data-driven tools to try to get at this question. But sort of to do this, you need some clear question. So we all know that humans are somehow going to impact disease dynamics, but to directly relate those, you need some type of biologically direct question where you can say this change in human dynamics, we can see that in terms of this in the pathogens. You also need a lot of data about both individuals and populations. And historically, data about large populations have been difficult to obtain, which hopefully now, with some new sources of data, we're able to combat that issue. And my research focuses on a very direct applied question about how human travel is impacting infectious disease dynamics. So this is a map of the spread of the plague in Asia and Europe in the 1300s. And historically, we've always known that humans are able to move pathogens around places that can lead to disease outbreaks in new populations. This has been a problem for a very long amount of time, but sort of what we do about that and how we're able to model, quantify, and predict that has been historically difficult. And the way human travel can impact spatial disease ecology is suppose you had these two different populations, one in orange and one in blue. And say you've never seen some type of pathogen in the blue location. Then over time, you see an outbreak of the disease in this orange population, and then later you see the disease emerging in this blue population. And so what could be inferred is that people who were infected in the orange location traveled to the blue location that led to a disease introduction that led to a disease outbreak. This is important to understand to try to identify if you should be targeting public health interventions or if the ultimate underlying dynamics may synchronize in some way as there's a trade-off, a trade between susceptible and infected people between these populations. So my research is sort of thinking about the human ecology aspect of it, and it sort of falls in three different parts, trying to look at more general public health questions, a focus on directly transmitted diseases, and vector-borne diseases. In public health, I've worked a lot about vaccination and about how people are traveling to health facilities and how long that's taking. For directly transmitted diseases, I've worked a bunch on rubella and measles and Ebola, trying to look at how human travel is impacting those. And my favorite are vector-borne diseases, looking at malaria and dengue, and how human travel impacts their disease dynamics. And a lot of my research tries to balance between sort of modeling and developing theoretical tools with data-driven projects and also field studies. So I work in a number of countries across Africa and Asia, shown in dark gray here, on predominantly directly transmitted and vector-borne diseases. In a number of countries, I'm running field studies with collaborators. So in Kenya, we've done lots of stuff on malaria and rubella. In Madagascar, we're working with the Institut Pasteur, looking at a range of infectious diseases and also people's nutritional status. In Pakistan, we've collaborated with the WHO and the Ministry of Health there, looking at dengue, polio, and measles. In Bangladesh, we're looking at dengue and malaria and the spread of malaria-resistant parasites. And then in Vietnam, I've worked a bunch on influenza. So today I'm going to talk about some of this research. So the first I'm going to talk about how we actually quantify human travel patterns. And then I'll give two examples, looking at disease elimination and control in the context of malaria, and looking at predicting disease outbreaks in the context of dengue. So I'm going to start with looking at travel patterns. So we've always known that people have been moving, and there's a long history of anthropological studies that are trying to identify how different populations are moving in space and time. So this is just a quote from a 1960s report that this guy who worked with the WHO to investigate sort of the role of human travel on malaria, where he's able to, perform an anthropological survey to see about, how things like the environment are predicting about where and when people are moving, and how far they are and when they're doing these things. And also in this report is sort of, we've been able to see this on both small scales and large scales. This is a map of sub-Saharan Africa, and there he's highlighted sort of the direction of different types of labor movements and why and where these people are moving. But to sort of make this actionable and used in modeling and predictions, we need to figure out a way to take these very detailed, very useful anthropological surveys and studies and quantify them in some way. And so quantifying these patterns also has a long history on various different types of temporal and spatial scales. So thinking about, where are people going during the day? Are they commuting? All the way up to how are different countries' populations changing through migration. And different spatial scales, thinking about very local travel, how many people travel within 10 miles to get to work, all the way up to international travel. And there's been a range of data sources that have been used very successfully in many cases, trying to sort of cover a lot of this space. So people have used work on GPS data to try to look at how people are spending their time locally, running travel surveys, asking individuals where they've gone in the last week or month or so, looking at international flight data, and the census data has been historically one of the best sources of mobility data, particularly for low-income settings. However, all of these data have some biases, as all data does. The census is only taken every 5 or 10 years. A number of countries have not taken a census ever, or they've taken one a really long time ago. Flight data likely represents only a small percentage of all the travel that might be relevant. In GPS data, it's very laborious to do these studies, so you can often only enroll some small number of individuals. And travel surveys, it's very difficult to design, sort of figuring out what question and how you're actually asking that question and how to make that representative over different areas. So if you ask people, how long does it take you to travel to work, that's going to get a really different answer if you're asking it in rural Kenya versus if you were asking it in Boston. And so my research, so ideally, what we want to be able to do is we want to take sort of this rich understanding of the cultural landscape and try to figure out how to quantify, individual trips for many, many people, where they're starting from, where they're going to, how long they're going there, and how frequently that's occurring. And many of these data sources sort of get at some set of those questions, but maybe have biases in other ways. But new data sources, hopefully, are going to be used to try to quantify human travel. But I think part of the issue with these new big data sources is how do you actually figure out what is epidemiologically relevant in this context? And so you just don't have more data, you have more useful data too. So lots of my research has been in developing methods and computational tools to process data for a science and policy relevant question. In particular, my research has used a lot of mobile phone data, figuring out how to extract mobility patterns and then identify the impact of travel on disease dynamics. So I'm going to briefly go over how we do this. So what we do is we partner with mobile phone operators, and we use their call data records, which are kept by the mobile phone operators for billing purposes. So if you have these mobile phone towers and you know roughly where they are in the country, for every subscriber in the data set, we can see a hashed ID and where they're making their calls or text messages, receiving them, and when that's occurring. So if we had this one individual, and suppose on day 1, we're trying to figure out, on each day in our data set, or it could be every hour or something, what tower are they using the most, and what location is that. So say we had this caller and was taking calls, making calls in this purple B area, then started making calls in this this orange C area, and they made additional calls there. What we'd infer is that their most used location that day was this orange C area. The next day, say they make a call around this A location that's green, and they make another one, and then we'd infer on day 2 that that individual most used tower A. Based on their locations on each consecutive days, we infer that this subscriber made a trip between that orange area and that green area. We can do this for every single day in the data set. And so this person, we'd see these different trips. So they made this trip between day 1 and day 2, a different trip between day 2 and day 3, and so forth. We'd see also that this person likely stayed in this orange area between day 4 and day 5. So we have an idea both of how many people are in a location and how many people are moving between all those locations. We're able to aggregate these mobility patterns up for often millions of individuals. So this is just an example for some of the data sets we've worked on, how many subscribers are in these data sets. So we have mobility estimates for 15 million people in a country over the course of the year, and from that we are able to get a rough general countrywide picture of travel patterns. So we're working with a number of collaborators across both in industry, public health, and academia in a range of countries to try to apply this type of method of extracting the data with mobile phone operators to apply to a range of infectious diseases. So some of the work that we've done is about correcting the biases in these data. So we know that not everybody owns a mobile phone, like little kids are unlikely to own a mobile phone, for example. So we've tried to do some work to see, given those biases in ownership, how do those skew our estimates of mobility patterns? We've also looked at how some simple models that are commonly used can be used to describe human travel, how they compare to other types of data like the census, and how do these travel patterns vary seasonally. So now I'm going to talk a little bit about how we've actually used these data in the context of malaria. So malaria is a mosquito-borne infectious disease caused by Plasmodium parasites. And so it has this complex life cycle between the mosquito vector, the human, and the parasite. There's about 200-something million cases of malaria and about 500,000 deaths. And it's important to note that you can be asymptomatically infected. So not everybody who has malaria experiences clinical symptoms. So it's possible for people to have the parasites in their blood and contribute to transmission without necessarily knowing that they're very ill. Majority of malaria cases are in sub-Saharan Africa. So this is a map of the Malaria Atlas Project, from the Malaria Atlas Project, that looks at parasite rate, with red being high. So many of the burden is disproportionately in sub-Saharan Africa. The map of malaria control and elimination has changed greatly. So all the countries in pink are ones that were controlling malaria in 1970, with very few countries, pretty much only Europe and parts of North America, that were considered malaria-free. Today, this map has shrunk a lot. Many more countries are controlling or have eliminated malaria. And as this map shrinks, we're going to have to start thinking, we might have to start thinking differently about control. So malaria control can sort of target any one of these components or some combination of them. So you can spray for vector control. You can have people sleep under bed nets that reduces the interaction between vectors and humans. There's a vaccine in development. You can increase people's access to drugs, which would clear the parasites. But why do we care about human travel? So there's two main reasons. One is about malaria resurgence, and the other is if we could be deploying our interventions in a more targeted way. So malaria resurgence is after you've sort of controlled the disease in some way, but there's still the possibility for onward transmission. It gets reintroduced by humans who are infected, reintroducing it into the environment. This has occurred historically in many, many different countries. And now, as the map of malaria shrinks, it's increasingly going to be a bigger issue if you're trying to control in a place. But if it's just going to come back, then that changes your priorities. The other idea is about how to target interventions. So this is work by Hugh that's looking at so the places in red are these transmission hotspots. And you have people who maybe live in the village, but they actually get infected in these other places like a forest or camping there or going to work. So originally, if you were planning on doing your vector control where you actually saw these clinical cases, you're not actually targeting where transmission is occurring. So if you know that these people, you see them in clinics here, but they're actually getting infected there, you should actually be targeting your control there. And so the idea is trying to figure out how humans are moving and how parasites are moving to see if you should be targeting your interventions differently. So the way we do this is from the mobile phone data, we have this idea of movement and connectivity between different villages in the country. And we're concerned about people from a high malaria risk settlement going to a low malaria risk settlement where they may be able to reintroduce the parasites. On the other side, people from a low malaria risk settlement might become infected here, and because they've never seen an infection before, they might be more likely to have clinical symptoms, and they also might be able to reintroduce the parasites. And identifying this is going to be key for any country trying to eliminate malaria, because it defines how vulnerable a place is to sustained elimination, and it can lead to these secondary things, secondary transmission, but it's very difficult to identify these importation estimates. And in particular, because we want to actually be able to measure all of these things at once. So what we tried to do is try to figure out where people are moving and where parasites are moving. So we first did this in Kenya. So this is that same parasite rate map in Kenya, where places that are in red are high. So there's sort of two clear hotspots of malaria transmission in the country around Lake Victoria and the coast. And what we do is we use a model to look at the flow of people from these high-risk settlements to low-risk settlements. And we measure the trip data using mobile phone data. And we're looking at people who may be reintroducing the parasites, but also experiencing new clinical symptoms and reintroducing parasites. So the mobile phone data we analyzed covered about 1 year in 2008-2009. There are about 12,000 mobile phone towers and about 15 million subscribers. And at the time of data collection, the operator had about 92% market share. And you can see that the mobile phone coverage is biased. So it is biased where people are. So you have the highest density in places like Nairobi and much lower in places like Turkana, for example. So some of the results are we identified these sources and sinks of travel and parasites. So sources would be places that are sending out a large number of people or parasites, and sinks would be where those are being received. So based on the yearly travel values from mobile phone data and importation values, so combining the travel data with a model and prevalence data, we're able to identify these sources and sinks. And so this is the source sink map for both travel and parasites. And you can see the sources and sinks of travel. People travel locally a lot, so there's a high source and a sink next to one another. The map of parasite, right, is the map of parasite movement is very different, where there's sort of a clear source around Lake Victoria and clear sinks in the Highlands region and Nairobi. We've ultimately trying to see if we can use these for public health planning. And to that end, we were collaborating with the CDC in Kisumu, that they were trying to look at the impact of targeted interventions. So the idea was that they were going to do a control program where they would target this one district, say a county, where they would treat everybody, and they wanted to see if you could see this spillover effect. So since we were able to identify, that area is where a lot of the parasites in the rest of the country are coming from, if we target that area, can you see an impact in the transmission over the country? And in particular, can we see this sort of very locally? So the mobile phone data has very fine spatial resolution, and can we also make these estimates for even different neighborhood blocks? Right now, we've been doing lots of stuff on malaria in Madagascar, so we're trying to look at a similar problem about the risk of malaria reestablishing on this high plateau. So Madagascar does not have any penguins, like the movie suggests, but it does have malaria along the coast without much in the center of the country. And so people are trying to understand, what's the risk of parasites moving from the coast inward. And with this, we're collaborating with the Institut Pasteur. And some other malaria work going on is across Southeast Asia, working with the Mahidol Oxford Research Unit to try to do a similar thing there, where you have a very different epidemiological context. So one issue is that there's been resistance to the frontline treatment found in these very remote forested areas. And it's not actually clear that mobile phone data will be able to capture these movements. Not everyone will, ownership is much lower there, tower coverage is lower too. So we're trying to figure out by running travel surveys, epidemiological surveys, and analyzing mobile phone data across many countries in Southeast Asia, how well do all these different things capture this high-risk population? And in particular, we're running studies to collect case data and blood spot, combining it with travel surveys and mobile phone data. So next I'm going to talk a little bit about predicting disease outbreaks in the context of dengue. So dengue is also a mosquito-borne infectious disease caused by the dengue virus. There are 4 distinct serotypes that circulate, and it's important to note you can be infected with different serotypes over the course of your lifetime. There's a huge number of cases likely per year, upwards of about 390 million. And the majority of cases are dengue, and there are some small percentage that are dengue hemorrhagic fever. The interesting thing about dengue is that it's actually emerging in many parts of the world. So there are some places, like Southeast Asia and Thailand, for example, where it's been endemic for many decades. But in lots of other places, it's emerging. So this is a map by Samir Bhatt that was in Nature that looks at the probability of occurrence of dengue, and you can see that the probability is much higher in these places. But this map is changing very regularly because infected people are able to bring the virus into new populations. And this is not an issue that's just about dengue. I mean, this is happening with many mosquito-borne diseases. There have been a very large yellow fever outbreak, and Zika is what all my relatives talk to me about Thanksgiving, which I don't actually work on Zika. But so what we wanted to do is we wanted to see how are we able to actually look at the risk of a disease being imported using these data sources. So what we did is we analyzed a dengue outbreak in Pakistan from 2013 using reported case data. So dengue in Pakistan, so Karachi is a big port city in the south of the country, and they've had dengue outbreaks for many decades. So the first really big dengue outbreak was in the early 90s. Every year since then, they've had really large outbreaks but fairly confined just to this port city. More recently, other parts of the country have started to have dengue outbreaks. So they think that the tire trade moved the mosquito vector across the country, and now that it's established elsewhere, every year people from the outbreak in Karachi are able to reintroduce the virus into different parts of the country. And so we analyzed one outbreak that lasted for 7 months, and it starts in March in Karachi, this port city, and then it spread through to Swat and Lahore. So Lahore is a big city in Punjab, and Swat is this more remote city up north. And in particular, we wanted to see, dengue is not a common, even though it's been endemic in Pakistan, it's only really been in that one city. So the idea, sort of the risk map for dengue in Pakistan is not well established. So we wanted to see what people had been using about this vector suitability. So, where could the vector persist? Can we figure out a way to refine this measure? Dengue is, there's no real treatment for dengue besides giving people fluids and trying to bring their fever down. So it's really important that doctors and public health officials are trained to be able to diagnose that people might be having dengue. And in many parts of the country, this has not been a problem. So they wanted to know if, can we actually use some type of mobility estimates to refine where they should be deploying particular doctors. And so what we did is we look at the susceptible population across the country. We include some environmental suitability measures for the mosquito vector, and then we look at the virus being introduced in the population by human travel. So in Lahore and many other parts of Pakistan, it likely gets too hot and too cold to have year-round transmission. So parts of the year, the temperature is such that it's unlikely the mosquitoes are able to sustain transmission year-round. So it's likely that the virus is being brought in by people. And ultimately, we want to see how all this works by predicting the case data. So our dengue model includes an SCIR type model for humans and then a mosquito component with interactions between the humans and the mosquitoes, and many of the mosquito parameters determined by temperature. We used mobility data provided by Telnor Pakistan, and this is a map of population density, so places in red are higher population density, and if places are shaded, those have mobile phone towers. So about 91% of the tehsils in the country have a mobile phone tower, and the subscribers are about 40 million over 7 months. And in particular, we noticed that there's really large amount of travel across a major highway that goes down from Karachi all the way up to Peshawar. So what we did is we used our human and mosquito model and looked at separate populations, and we used the model to fit when the first introduced case occurred. So we have one location, and we see this, in orange, we had this disease outbreak, and it was likely that the first introduced case occurred then. And this is inferred only from the model and the case data. Then we try to use information about the dynamics in other parts of the country and the connectivity in other parts from mobile phone data to try to predict if we can estimate when this disease introduction occurred. One particular thing to note about mobility in Pakistan is it didn't seem to follow this normal decay with distance that's assumed. So lots of people assume the further you are away, the less likely you are to travel there. In Pakistan, probably because they have this major highway that connects all the economic hubs, you actually see a large amount of travel quite far distances. And we find that impacted where we thought the disease was going to be spread to. So this is the actual case data, and these are some predictions with the mobile phone data and with that diffusion model that has a strong decay with distance of when you're going to have an imported case. So places in red would be places that had an early imported case. Places in blue have a later one. And you see that the majority of cases are actually in this northern region of the country, with very few cases actually reported in the southern Pakistan. And we see the mobile phone data is roughly able to capture that spatial trend, where you see a large amount of early introductions up into Punjab and north, whereas the diffusion model here is predicting a large amount of cases in southern Pakistan that historically have not reported many cases of dengue. Given the granularity of the data, we're actually able to look at a much finer spatial resolution. So this figure is a little bit confusing. So what we did is we took particular tehsils in the country, and we tried to estimate, just based on the case data, when did the first case occur that led to this outbreak. And there's a lot of sensitivity about is it really one case, is it 10 cases or whatnot. But sort of when were there enough cases that were being imported into that part of the country to lead to this outbreak, these case data. And then so the gray here is the vector suitability. And so you see that around a lot of the year, you could have had an outbreak, but we estimate that sort of the case that led to this outbreak occurred around here. And then what we did is we compared this to our predicted introductions from either the diffusion model or the mobile phone data. And so here, anytime there is a circle or a square, that's when there's been a predicted introductions, either from that diffusion model that has this decay with distance, or from the mobile phone data. So in Lahore, which is the city that had a very large outbreak, the predicted introduction from the case data occurred around here, and we can see that both the mobile phone data and the diffusion model are able to roughly capture introductions in that time span. The mobile phone data would predict introductions a few weeks before than the actual first case that led to the outbreak. But for public health planning, it's much more useful to have sort of a lead time here. So this is able to say that both data sources, so even if we hadn't had the mobile phone data and just use the diffusion model, you're roughly able to predict introductions, but also that the mobile phone data predicts introductions earlier. We also looked at a number of other places. So this is Swat Valley, that's this one particular city. And 2013 was the first time that place had ever reported any cases of dengue. And we did a similar thing, where we looked at the actual case data, and from the model and the case data, we predicted when it was likely that the first introduced case occurred that led to this outbreak, taking into account also the vector suitability. And in this, we actually would not predict any type of introductions from the diffusion model. Swat just seems to be too far away from Karachi that it seemed unlikely, based on the model, that there'd be any introduced cases. Whereas the mobile phone data is actually able to predict some introductions right around this wide band of when we think the first introductions occurred. Then what we did is we tried to use this vector suitability map to combine it with these importation estimates. So every day in the data set, you have sort of, is it possible to have disease, mosquito transmission that day, but also are there any importation estimates then too? And so if it's possible to have transmission but no importation estimates, that's a low risk. But when those two things are going together, when it's likely that you could have transmission and there are importations, that's when you'd want to be able to see that there's a higher risk. And so using that, so the mobile phone data and the model, we were able to look at sort of the epidemic risk and refine this type of map much more. And I don't have the results here, but for this map, we also were able to look across time. So we could say in particular parts of the country at particular times, when is it likely that this place is at an epidemic risk or not? So we're currently working with the Ministry of Health in Punjab and the mobile phone operator to try to figure out if this can be used in sort of real time for disease risk mapping tools. So I think the dream, which we're far from at the moment, is that the data, because we worked with the mobile phone operator there, so all the code runs on their servers, that they could process all the data and get out these aggregate mobility matrices that could then go directly to the Ministry of Health in Punjab that they can use for a range of public health purposes, not just dengue. And that they could also, we are working with them to try to figure out how to develop a tool that sort of directly feeds into their case data, so they know that they have an outbreak of disease X in this location, but we can say, based on some a simple model and the mobility data, if you see the case X here, where might it be going to? And how can you use that to try to plan different coordination strategies? So there's a lot of ongoing work now with dengue transmission, and this is predominantly with Henrik Salge at Hopkins and Institut Pasteur and Derek Cummings, of trying to look about on much finer spatial scales and relating it to genetic data too. So most of the work we've done has been on this large country population level mobility. We're looking at people who travel, 100 miles between places that have very distinct disease outbreaks. But the mobile phone data, we presumably can look much finer too. We can see, people moving around different parts of the city over the course of the day. But it's unclear exactly how that can be related to different disease processes. So dengue, so we're working with dengue in Bangkok, and we're looking at different types of case data and genetic data to try to look at different transmission chains and how those transmission chains are related to how mobile different populations are. So this is a map of Bangkok. It's a little bit hard to see, sorry. And places in blue are places that are not as mobile. Those populations don't move as much. And places in red and yellow, those places are moving much more. And so is it true that places that sort of have a high genetic diversity of transmission, high genetic diversity of different types of dengue viruses, are those places where those people are also really mobile? Is that true, is that not true? Or is everybody sort of getting infected in sort of the same local area? So this may not work at all, but we're trying. And we're also trying to see sort of if you can look at how many steps away two viruses are. Can that relate to how far away they are by physical distance or travel? So we have mobile phone data from Thailand, and so we're trying to see how well these different things are relating, both for Bangkok and looking at these fine-scale mobility patterns, and for the whole country. And so I think I talked very quickly, sorry. But sort of roughly, yeah, I'm interested in thinking about human population dynamics and how that can be used for different types of vector-borne disease ecology and public health. And so I'm just going to briefly go over some of the ongoing work. So with Catholic Relief Services and some people at Harvard, we're looking at sort of nutrition and disease incidence to see about the incidence of childhood infections and people's nutritional status. So Madagascar is historically one of the top countries for stunting. And can you actually observe sort of different people's nutritional status? Does that have an impact on disease dynamics? It may have none. It may have some. We're not really sure yet. We're also deploying a number of vaccination coverage surveys in Madagascar with a Sierra Leonean medical doctor trying to look at how people are actually getting vaccinated in the country and if that would change what we put into our disease model. With some collaborators at Southampton, we're looking at seasonal estimates of population. Some parts of Madagascar and other countries, no one lives there part of the year, and the rest of the year it's a complete, it's a small city, and trying to see how that changes our estimates. For directly transmitted diseases, I'm working a lot on thinking about metapopulation rescue effects of childhood illnesses, and in particular how that changes as disease control and elimination changes, and running some serological surveys of looking at people who are co-infected. And for vector-borne diseases, which I talked about a little bit more, of looking at just broadly malaria control in different countries, how well do the methods we used in Kenya apply to different contexts, looking at travel surveys and sort of these fine-scale mobility and transmission chains. And to continue try to figure out, how can we integrate these field studies, data, new types of data, and modeling studies too. So some conclusions. So human travel can impact disease dynamics by introducing the pathogen into susceptible population. There are these new data sources, like mobile phone data, that we can use to quantify human travel on fine spatial and temporal scales. But sort of trying to figure out, how can we quantitatively understand human ecology? And how can that be used, with more traditional epidemiology and ecology and mathematical models to ultimately guide public health policy and planning. So there's a very large number of collaborators, including Caroline Buckey, who's at Harvard, and Jess and Brian, who are at Princeton. And this work has been funded by a large number of people, including the Burroughs Wellcome Fund, which I'm currently funded by. Thanks.
Two questions.
Yeah.
First is, what is the length scale of the distance between towers, cell phone towers? Is it kilometers, tens of kilometers?
So it varies.
Yeah.
So some places you'll have multiple towers on the same city block, and other places it'll be 10 kilometers away. But in general, a mobile phone tower roughly can cover up to about 3 kilometers.
Okay, so that's the resolution.
Yeah. So it's hard to measure further than 3 kilometers with one mobile phone tower.
Okay. And then the other question is, can you say a little more detail about how you infer the things that you're not getting? Like you said, children don't have cell phones, but you know they're going to school or something like this. But how can you use the data that you have to inform that or
Yeah. So I think the fact that kids don't have phones is disappointing. I mean, maybe not for humanity, but for this, yeah. So
we should tag them all. We should imprint it.
I'm not doing that, IRB.
Okay.
So in some things, what we've done is we have compared it to travel survey data, where they ask grown-up people, how often do you travel with children? What ages are your children that you're traveling with? To see if we measure it's 100 people who are moving, how many kids might that be? And in particular, does that change where we would say the primary sources and sinks are in a country, for example? So in general, we find that kids aren't traveling as much without adults. So it's likely that we're overestimating the importation estimates, but we're roughly getting the same routes of travel correct. But I think, thinking about how general are these countrywide patterns that we're measuring and how biased are those depending on which type of pathogen. So in particular, I haven't worked on HIV because then you care much more about the population level heterogeneity, where this data, because it's very coarse and very general, is likely missing a lot of those things that are going to be relevant for transmission. So sometimes we multiply. It roughly looks the same color scale, but I think it's important for depending on the pathogen.
Let me ask one follow-up question. I think we have time. When you have the raw data, and how much do you massage it? How much do you have an underlying model that is putting some more assumptions into it to really get your final mobility information out?
So in terms of just extracting the mobility data from the call data, so we don't have an underlying modeling assumption. Our assumption is, that wherever you were the most that day is representative of where you spent your time. We do assume that if you made a call on this day, you didn't on this day and this one, that you're where you were most recently. So we do fill in that part, but we don't have an underlying assumption that people can only move a certain amount or that people are likely to only travel to this many places. We pretty much just process the data, smooth the data a little bit to get rid of some of the heterogeneity in calling behavior, and then use that as it is.
To what extent do you account for population heterogeneity? So, some subgroups in the population are traveling more than others or may have different, may be at risk in different ways based on socioeconomic status, race, etc.
Yeah, so we are, so for certain pathogens, we haven't necessarily taken into account sort of biases in at risk. So dengue, like lots of people can get dengue. In places where it's endemic, there's a much more socio-demographic gradient. But in places like Pakistan, where it's emerging, wealthy people, accessing care is different, but lots of people are getting affected across different income brackets. I think we're trying to see how well these sort of general mobile phone data patterns are representative of very at-risk populations by running these travel surveys in Southeast Asia. So for malaria in Southeast Asia, it's highly biased, so it's a very particular populations that historically have not been able to receive care or are migrant communities. And so there we know that the mobile phone data is likely overestimating. We're not sure exactly by how much, but that's why we're trying to do these epi studies too, to look at who is actually being captured when we use these other data sources and what risk factors are we missing.
So you mentioned that the Kenyan CDC picked up on your work and said a targeted intervention there along the Lake Victoria shore. I wonder how precise, did you make very precise predictions about what the outcome of that intervention would be, and if so, or more generally, what's happened since?
Yeah, so we did not make very precise projections of what would happen.
Why not?
Because I don't think people know what's going to happen when you do these mass drug administration programs.
Doesn't your model can make predictions?
So our model is able to identify places where you're likely to see an impact. So if you want to set up a particular surveillance site to try to catch these people who might be targeted in this type of, so they live here and they get a special, a course of drug treatments here, and then they move to other parts of the country. These people tend to move to these parts of this part of the city or this part of the country, not necessarily what impact that will have for onward transmission. So I think there's, it's still unclear exactly how effective these mass drug treatment programs are. And so I think that uncertainty would make would not necessarily make our predictions very accurate or as useful. We're just trying to identify where increased surveillance might be useful, not necessarily it should be a reduction in X number of cases. Because I think it's still unclear, when you do an MDA, how many cases does that reduce even locally?
So more generally, so what are the prospects then, do you think, for coming up with falsifiable predictions that would allow you to know whether your estimates of human mobility really are a key factor?
So I think we're trying to do some stuff, so like the dengue stuff, we predict imported cases in lots of parts of the country that have never reported any cases. So is that true? Is that true that there's no dengue cases there? or is it true that no one diagnoses dengue there? So we worked with the Ministry of Health and gave them more detailed estimates about where we were predicting introductions to try to do some investigations to see if there are actually an underlying dengue outbreak that's just not getting picked up by traditional surveillance. Yeah.
You showed a map of occurrence across the world at two different time points. So when you're thinking about longer term changes in prevalence or occurrence spatially, how do you separate out the relative contributions of interventions versus changes in travel patterns?
Yeah, so I think it's going to depend on what pathogen you're looking at. I mean, it's, in most things in public health, just improving people's access to care will do much more than any of the rest of this stuff. I think trying to figure out, when you have an emerging outbreak, what places might be hit next in sort of a shorter time span seems to be a more useful utility of these kinds of data, as opposed to trying to project for these really long trends. How are things like if you increase the community health workers in a place versus you were able to identify imported infections? I think that's harder. Yeah.
I wonder whether you can comment on differences in utility of this approach for vector-borne versus directly transmitted diseases. Because part of me is thinking, well, it could be that the vector-borne transmission is happening below the scale of your data, right, within this 3-kilometer thing, and so you're capturing this other scale of data that you can't really get to. But it also, another part of me is saying, well, you could also tear apart the tie between human mobility and disease transmission. You've got this other vector thing going on. So yeah, just wondering.
Yeah, so I think this is a really good point. Yeah, some estimates of dengue transmission think that most people get infected within a kilometer of their household. And so I think, and that is a scale that is very noisy with mobile phone data. So I think we're trying to sort of capture broad patterns for vector-borne diseases and where the disease might be introduced and established there. And then if it becomes endemic there, I think that becomes then a question of the local mosquito biology more. For directly transmitted diseases, I didn't present any of it, but we've predominantly tried to look at sort of the seasonality in travel patterns and how that relates to seasonality and transmission. But I think for many endemic diseases, it's not as important to understand the travel patterns. It's more important to understand the endemic dynamics. And travel becomes more important when you have a very heterogeneous control program. So if you're only deploying the vaccine in certain places, or you've only vaccinated in certain places, is there a way to target that vaccination program, or is there a way to identify where there might be risks that if you definitely miss this place, it's going to be introduced or something?
So the method you're developing and this general approach you're taking to harnessing mobility data and using it to answer questions about disease transmission and so on, I wonder, have you thought much about other problems or other diseases, maybe other things which don't involve diseases, to which these same kinds of methods might apply, and if so, where do you see opportunities?
Yeah, so I think the other area that a lot of these data have been used is about disaster response. So when you have a hurricane or an earthquake and lots of people leave, can you predict where they're going to? Based on who they're calling most frequently and where they travel and that sort of stuff. So lots of our collaborators have been sort of pushing that avenue much further. I think in terms of, does that answer the question?
That's, yeah.
What was I feel like there was more.
Yeah, just wondering if you thought beyond the sort of infectious disease context or outside of the cell phone context, even just thinking about other sources of data or other systems to which these approaches might still be useful.
Yeah, so I think more generally thinking about how introduction events impact local dynamics and how different settings for disease prevalence or endemicity when in and how immigration or extinctions could be impacted. I think we've tried to do it in a very like focused, for malaria control in this part of this country. But I think generally thinking about those kinds of questions, I think is really interesting. Most of the time though, I tend to either think about mobile phone data or diseases, or some combination of them. But we've also tried to look at sort of more general mobility patterns too. How dynamic are different population estimates? How does that impact your model? How are people accessing healthcare seasonally? Does it vary a lot when you have a road washout? That sort of thing, which I think is sort of maybe just like a cousin, but not necessarily a different lineage. Mm-hmm.
Yeah, I have a question about whether you have any predictions about what is the spatial scale of human travel, how that impacts the evolution of the pathogens in terms of birds. So, what do you expect if you have large-scale distance versus local-scale distance?
Yeah, I think that's a really interesting question. I think it's going to depend on what pathogen you're looking at and sort of what that means in terms of how fast the pathogen can evolve on its own and how much pressure from other distinct versions of that pathogen, what that means in terms of its evolution and stuff. But I think, yeah, it's a really interesting question, yeah. So I think my answer is I don't really know, but it's probably going to depend on the pathogen. Mm-hmm.
Just kind of thinking about taking this forward, going back, I'm curious how many years back this data is available, and what plans are for taking it forward. It'd be interesting to see how some of these patterns change with economic development, and what could be done to, I guess, like work with future planning as opposed to always being kind of in response to stuff, and then you could look back and see what happened to bring this here.
Yeah, so I think, yeah, it's a really good point. So the longest data set we work on is about 4 or 5 years of data, but it's continuing, we get new data every 6 months or so. And so what changes you're able to see on that time scale might be difficult in 4 years, what's changed or whatnot. I think some of our collaborators have worked in Haiti a lot, which has had like a number of natural disasters, so then you've seen a much bigger change in sort of the population structure, mobility structure in that country, even over a few years. But yeah, so I think so that's sort of looking back and seeing what has changed. So then going forward, we're trying to figure out, how the data could be organized and processed very quickly with the mobile phone operators, that then they could be used in sort of almost real-esque time is the goal.
Traffic. Red spots on the highway.
So I think people have done that a lot in the U.S., looking at travel patterns and stuff for traffic and sort of city planning and things. But yeah, I always like diseases more. Yeah.
That is sort of a specific question. You said that the travel data is based on phone calls, spoken phone calls, or it's just any sort of phone tower connection.
So it's not data, but it's voice or text message.
Oh, okay.
Yeah.
All right. That's it.
Yeah, so it's not like smartphone data. These are all like the Nokia phones with a flashlight, that level.
Very Heisenberg's survey principle. But using the phones, has it affected the mobility of people themselves? I mean, if you can contact somebody remotely, you may not have to travel. So potentially the actual rate of traveling has gone down with the adoption of these technologies.
That'd be unfortunate. Yeah. So we're also trying to figure out, like if people are traveling, are they more or less likely to use their phone or not? To see also about those sorts of biases of things.
I was wondering whether there were any more general patterns that you could bring back to other kinds of organisms other than just humans and human diseases, sort of things you might infer about as you improve your models to study dengue and you understand how rates of movement influence of dengue, can you bring it back to other kinds of wildlife or monarch butterflies or any other kinds of organisms?
Yeah, I think sort of generally thinking about these, what kinds of models you're using and what assumptions are you putting in your models and and we see, like what well mixing does to your model. And we, can say in parts of the country, they seem to be less well mixed than others, so maybe this type of thing, which could, I guess, be generalizable to other types of pathogens and that sort of thing. Anything that moves.
Doesn't
Over
have to
some,
carry a cell phone.
yeah, over something that you can make a map of, ideally, I think.
So you focused on extracting this data to find movements of individuals, but have you thought about, or have you used the pairwise nature of this, looking at contact networks, especially for directly transmitted infections where that contact network structure matters? To figure
Yeah.
out
So,
who's gone who?
yeah, we started to look, when I originally started this, I originally started this project when I was an undergrad at SFI, and then we were trying to compare between sort of contacts structure and mobility structure and how those were the same or different. But I think then I sort of got much more focused in understanding for very specific diseases where there's this much more spatial component than contact component. But I think, yeah, thinking about what contact structure says about these, I think it's often difficult to get disease data to be able to fit to and to be able to calibrate with on the same types of scales that we're working on. But I think, yeah, thinking about, it's like an obvious thing that we should start be doing, thinking about that aspect too.